Good afternoon, everyone. On behalf of the BioTechMed Center, I would like to welcome you to this, the Feast of Neuroscience. We're going to have two amazing lectures by outstanding speakers that visited our Gdansk Tech University. I'm Dr. Michał Kucewicz from the Department of Multimedia Systems, Faculty of Electronics, Telecommunications and Informatics. And today, we're going to have two talks. First of them starting with Professor Milan Brazdil from St. Anne's University Hospital, which will start right now. And after that, Professor Greg Worrell will be from Mayo Clinic from the USA, will be having his talk. And around 3 p.m. we'll also open a grand discussion. So we have here a live transmission through a YouTube channel. So we welcome everyone who attended and is streaming right now and watching this transmission. We have the open chat available, which we will use later for your questions, for the debate. And there's two things that I need to mention. So one thing is that the contents of please do not record those talks. The contents of the presentation, some of them were not published and are very new. We will be talking about very exciting things, which are almost like from a science fiction movie. And about very exciting things, so please do not record anything. And the second appeal is not to ask questions during the talks. We will have time for questions after each talk, specifically for those talks. And at the end, there's going to be a grand discussion for any topic really. So today we're going to be touching on those interesting questions which are from the boundaries of neurology, neuroscience, psychology, biomedical engineering, cybernetics, and even touching on some more philosophical issues even, or from the realm of humanities. So think about those questions for the grand discussion, because we would invite any questions, and if you are from another discipline, please don't feel discouraged. We really want to address those talks to a general open public, to any scientist or non-scientist. So if you've got a question that you think is stupid or naive, please do not hesitate to ask it, because we're looking really forward to a big discussion here. So before we start, just a few words about our first presenter, Professor Milan Brazdil. So I have known Milan from the start of my postdoctoral research at Mayo Clinic. I think we first met right after I published my first brain paper, and he approached me on a conference. I was someone who was totally not known in the field. I was just, this starting postdoctoral researcher, and then Milan approached me when I was showing my poster about the high-frequency oscillations, and I remember we had a very stimulating discussion, and he impressed me as a person who would be like a real mentor, who would, come to a person who is like nobody. He was like, an established professor, and have like this discussion that really, really inspired me. Milan has got a lot of international experience. He's worked in the UK. He started the collaboration with Mayo Clinic, which you'll hear about more in the presentations. And has been really a visionary person. I think that the most important thing, just forgetting about going through CV and all your education, which is very impressive, I would say that the greatest thing about your research is that you made me think about questions that people don't dare to ask in neuroscience, like the déjà vu experience that you will hear today, or how the brain makes errors. Or his research also touches on issues of empathy, for example. So those are the questions which previously would not be addressed by neuroscience research, but clinicians like Milan have the courage and the passion, I guess, to address these questions. So I'm really excited and looking forward to hearing the talk today, and the floor is yours, Milan. Thank you.
Thank you very much, for very kind invitation. Working? Perfect. In this my talk, I would like to focus on something which is probably in Polish błędny mózg. erring brain. It's a fantastic topic, I think, and I would like to share with yourself my view on the errors in the brain, because I'm sure that we have to understand the errors in the brain when we want to understand how the brain is functioning. And without this info, very likely we will not be right or understanding ourselves even. So what does it mean, the errors? In general, error is, there is a lot of definitions, of course, and one of the definition is that error is an action which is inaccurate or incorrect, and very simply said, error equals a deviation from accuracy or correctness. When we are speaking, however, about the human brain and the human errors, then the definition probably should be much more elaborated, and according to James Reason actually monography, and which was published in Cambridge 1990, an error is the failure of a planned sequence of mental or physical activities to achieve its intended outcome when these failures cannot be attributed to chance. And what is extremely important in this definition is the inclusion of the word intention, because we've got some intention, and if the result is not as was intended, then it might be an error. And what's also extremely important, I guess, is that the human errors can be broadly distinguished into categories. And one category is category 1 errors, which is error of execution. Usually we are speaking about lapses or slips, and they occur when the action conducted is not what was intended. And the second category 2 error is error of planning. Usually then we are speaking about a mistake, and this is the situation when the action proceeds as planned but fails to achieve its intended outcome because the planned action was wrong. This is a problem with plan. In our everyday life, we are doing a terrible amount of mistakes, and there is a lot of publications focusing on the human mistakes. We are doing mistakes when our intention is to lose some weight. We are doing mistakes during the presentation. There is a lot of publications how not to do the mistakes. But in this my talk, I would like to rather focus on the category 1 mistakes, on the slips and lapses, and usually on the slips. One of the experimental tasks, which is very widely used since 1965 in the cognitive neuroscience research, is so-called oddball task. During this oddball task, there is a series of two types of stimuli. One type is very frequent. It's not so interesting. For example, the letter O, or the sound of some 1000 hertz. And in between there is sometimes, very rarely, presented another kind of stimulus, which is usually called as target, and the task for the tested subject is to respond in some way to these target stimuli. For example, by pushing the microswitch button in his or her hand, or to count these target stimuli. In 1965 already, Samuel Sutton for the first time found that if you average the EEG responses to after the target stimuli and after the frequent stimuli, you will get a clear-cut difference. And the difference is lying here in the presence of this very broad positive potential electrophysiological phenomenon, which is called as P3 or P300, which can be observed only after the detection of the target stimuli, of this rare stimuli. And it's practically missing after the frequent stimuli. This is a fascinating phenomenon which was studied for many years, and especially at the scalp recordings. And this phenomenon, this P300, is probably reflecting all the sets of mental processes, including discrimination, including the memory, including attention, etc. But after having the possibility to record the EEG activity, not only from the skull, but also from the depths of the brain, and this can be possible because of the epilepsy surgery programs, and many epilepsy centers worldwide has the possibility to record the EEG using the depths or intracerebral electrodes in patients who are intractable to the pharmacotherapy and who are the candidates for surgery. And prior to the surgery, it's necessary to clearly localize the epileptogenic focus, and after that, these focus can be removed to cure the patient. During this invasive EEG, usually a couple of electrodes, up to 20 electrodes, are implanted into the brain, to the different brain sites, and some of these electrodes are then usually localized within the epileptogenic focus, some of them outside the epileptogenic focus, and during, let's say, 5-10 days, when we are waiting at the epileptic monitoring unit, we are waiting for the seizure, which is absolutely necessary to have prior to analyze the data. We can perform also some simple cognitive task. And one of the tasks which was repeatedly performed is the oddball task. And what the data can tell us. Depth ERP recordings were repeatedly used to identify areas of the brain involved in the generation of different cognitive processes. So because we are, for example, recording from hippocampus, we can have a very good spatial information about what's happening during this discrimination of the target stimulus. And if there is, and this is extremely important, if there is no activation during the frequent and activation during the target stimuli. And besides, also, we can elucidate the temporal sequence at which different areas of the brain get involved. So the temporal information is another extremely important information which we get from these depth recordings. As you can see here in these figures, there is a typical finding from two contacts which are located in the anterior hippocampus in the patient, and you can see that in the first contact there is no P3 waveform, whilst in the adjacent contact there is a very nice potential, which means that there is the source of this potential. So we can say that definitely mesiotemporal region and hippocampus is participating in the discrimination of the frequent and target stimuli. So this is something which is telling us a lot about the functioning of hippocampus during this very simple task. Another sources are in the anterior cingulate, in the rhodolateral prefrontal cortex with this phase reversal, and this is another our paper published quite many years ago, showing that there are, of course, in hippocampus, for example, there are generators of task non-specific P3, so it doesn't matter whether the patient is pushing the button or whether he or she is just mentally counting the target stimuli. In both conditions, in hippocampus, there is very nice activation, and this phase reversal, which is again showing us that there is the generator or source of this potential. And in other parts of the brain, you can see, for example, some activations of movement-related potentials only if the patient is pushed to the button, and nothing similar when he or she is mentally counting the target. So we can have the information not only about these discrimination, but also about the action which was or is done by the patient. When we have these depth EEG recordings, and when we are able to have a look into the brain using these depth electrodes, what we did is also to study the unconscious discrimination of these frequent and targets. Again, more than 20 years ago, we presented to the subjects with depth electrodes not only the frequent and targets, which were very nicely seen and clearly discriminative each to other, But in the same experiment, there were also the stimuli which were presented subliminally, which means that the patient was unable to discriminate consciously between the frequent and target stimuli. But still, similarly to the situation with the supraliminal classically presented stimuli, when the patient is able to produce, or some part of the brain, in this case the orbitofrontal cortex, is able to produce P3 waveform after targets but not frequent. And after subliminally presented, still there is clear-cut difference in the orbitofrontal cortex, which is amazing. The patient is not able to discriminate, but his brain is. This is another example of the recordings after the subliminally presented, which is in the black curves, and after subliminally presented, which is in the red curves. And you can see that in the same place there are the generators of the same process, even if it's proceeding actually subconsciously or subliminally. This is another example. And you can see that in some brain parts, for example, in the left-sided dorsolateral prefrontal cortex, if the patient is able to discriminate consciously, there is the potential. But after subliminally presented frequencies and targets, there is no activation, whilst there is the activation in the cingulate cortex on the same side. So it seems us that in the brain are some parts of the brain which are extremely important for conscious or aware or unaware discrimination. And using this very simple task and simple tool, actually, we are able to tell something more about the consciousness also. When functional MRI came to the neuroscience research, there were a lot of researchers which wanted to replace all these very invasive techniques like depth EEG recordings with functional MRI. And also oddball task was quite heavily investigated using functional MRI. We did something similar, but we did the investigation of the auditory oddball task one day with the functional MRI, and 2 days after that, after implantation of the electrodes through these depth electrodes. And as you can see here, this is the data statistical parametric maps from the functional MRI. You can see that there is a statistically significant activation after the target stimuli when compared to the frequent stimuli in different brain regions, which seems to be very similar to what we knew already from our depth EEG recordings. But repeatedly, we were able to find this bold signal increase in different parts of the brain, and in the same patient, at the same moment, there was nothing in the mesiotemporal regions, while the depth EEG recordings clearly revealed the activation there. So it seems that some portion of neural activity which is evoked by target stimuli might be detectable only with EEG and might not be observed using the fMRI methods, even if this study is already quite old, and of course there were some progress also in the development of fMRI. But still, from my viewpoint, both methods should be viewed as complementary and not as interchangeable, actually. So, but come back to the errors. During the classical oddball task, the subject should respond to the target stimulus. And problem is that sometimes he could push the button not only after targets, but make a slip and push the button after frequency. And if, yeah, this is correct, and this is incorrect. And if we then average the EEG signals after this response, which might be correct or incorrect, which is erroneous, after incorrect or erroneous activities at the scalp and also in the depth electrodes, we can see something which is called error-related negativity and error-related positivity. This is after the incorrect and this is after correct responses. And again, we can find that in the brain there are some generators of these error-related potentials in some regions. And but this is not a typical example how to study the performance and errors in the neuroscience research. In a parallel to classical oddball task, there is existing go-no-go task, which is very similar, but only the instruction to the subject is completely different. The instruction now is not to push the switch to button after rare, but after every frequent target. In this task, it's much easier to do some slip actually. And we investigated, and the question is, this is something very important, because before these depth EEG recordings, this error-related negativity was exclusively recorded from the scalp. And using dipole modeling, the researchers thought that the generator should be the anterior cingulate only. But when we investigated these go-no-go task in 7 patients with depth electrodes, and in total we investigated more than 570 sites in different parts of the brain, and we could, in grand average, from these seven patients, there is a typical error-related negativity seen here. But in different parts of the brain, we were able to find the generators, not only in ACC, anterior cingulate cortex, but also in the medial prefrontal cortex, in SMA, for example, but also outside this mesial wall or the frontal lobe, including middle temporal gyrus or hippocampus, which seems to be active in all the tasks in general. This is another discrepancy which can be shown between the ERP, depth ERP data and fMRI data. This is the meta-analysis of 8 event-rated fMRI studies of error processing, and you can see the activation in this medial aspect of the frontal lobe, but no activation in any of these studies in the rostral part of the anterior cingulate. But we repeatedly found there the generators. So again, there is something which depth ERP recordings can very much help us in understanding the brain. So the question is, why does the human brain make mistakes and error? And it seems that if we are speaking about the mistakes, about the category 2 errors, it's clear that it makes sense to do errors, because we learn from mistakes. Correcting is key to learning. These mistakes help us to gain experience, which later serves as a blueprint to complete the same task successfully. So they are important and meaningful. But as I said, I would like to focus on these error 1, or category 1 errors, on these slips and lapses. In brain, very likely there is quite a lot of these slips and lapses, and one which might be seen as one kind of slip is this optical illusion, which is very widely known. This is Adelson checker shadow illusions. It's some kind of error in the visual system. If you compare the brightness of this square A and B, it seems that they are completely different. But the opposite is true, because you can see that actually these two are practically same, and this illusion is resulting from our experience of seeing objects in shadow, and so we expect something. And what's extremely important when we are actually evaluating the function of our brain is that everything must be taken in the context. And this is one very nice example how important is context. In all very complex systems or highly organized system, there are some errors, category 1 errors. It's very well known that in the construction projects there is approximately 7% of errors, and usually the designers has this issue in their mind. A factory produces up to 5% of defective items, and if the system is more complicated, like emergency department, the amount of errors might be up to 30%. I'm sure that everybody of you met on your screen this report, which makes us furious, and this is just computer. It's not so complicated as our brain. So sometimes we say that healthy brain is perfect. It's almost perfect, but still there must be some internal errors in the brain. Now I would like to tell something you about the déjà vu, which is an airy phenomenon, psychological phenomenon, which attracts the attention of many people from the 4th century, actually, because for the first time was described by St. Augustine as a false memory some 16th centuries ago already. It's a illusion déjà vu, widespread, fascinating, mysterious human experience, airy experience, in which we recognize that the situation is familiar, sometimes intensely so, while we are concurrently aware that this sense of familiarity is inappropriate. I'm sure that the majority of you knows this and experienced this déjà vu already. In the last century, this phenomenon attracted interest also of psychologists and in the last years of neuroscientists, which is not surprising actually. On the other hand, it's extremely difficult to study this phenomenon because we cannot evoke in healthy subjects these phenomenon. We know very well déjà vu in the context of the temporal lobe epilepsy, and we know that up to 30% of patients suffering from temporal lobe epilepsy are referring that regularly or sometimes in the beginning of the seizure as an aura déjà vu can occur. We also know that electrical stimulation of different parts of the brain in these epileptic patients can provoke this aura, this kind of aura, déjà vu aura. And there were several authors publishing the data, and it's not just the stimulation of one single structure in the brain which can provoke the déjà vu phenomenon. It was the stimulation of amygdala, hippocampus, rhinal cortex, or entorhinal cortex, but also some parts of the brain which are even more distant. This is another very important slide. I guess 9 years ago, Fabrice Bartholomé from Marseille published this very interesting study in which he and the colleagues showed that first of all that déjà vu is more frequently provoked when the entorhinal cortex is stimulated. And what's extremely important is that only if there was a correlation of the signal between amygdala and the hippocampus, or rhinal cortex and the hippocampus, then there was clinically occurring the déjà vu. So it's not about the stimulation of one structure, but about the communication between entorhinal cortex and other parts of the mesiotemporal region. This is important to know because when investigating such a complex phenomenon like déjà vu is, definitely we shouldn't focus on one single structure in the brain. It is also very well known that the stimulation of the lateral temporal cortex, and namely upper temporal gyrus, can provoke the déjà vu phenomenon, whilst the stimulation of middle or inferior temporal gyrus never will provoke this. And the explanation is that there is very heavy connections between the upper temporal gyrus and parahippocampal gyrus or rhinal cortex, whilst there is no such a direct connection with the middle or inferior temporal gyrus. So what's also known very well that in some psychiatric patients, the déjà vu may occur quite frequently in patients with depression, with schizophrenia, but even the neurotic conditions. But from my viewpoint, which is much more important, is that occasional déjà vu is reported in up to 80% of completely healthy subjects. And this is strange, because is it normal? Is it something which can be seen as a normal phenomenon? Absolutely not, but yes, because the majority of healthy people knows it, so it should be normal. And now the problem is, why does déjà vu happen in completely healthy subjects? There is a number of hypotheses. Starting from past life memories because of reincarnation, astral teleportation, and many others. There are more than 30 psychological or neurological hypotheses, but frankly, still there is no widely agreed explanation. What does it mean in healthy subjects? But more than 50 years ago, Wilder Penfield wrote, déjà vu might be an ictal phenomenon in normal individuals as it is in patients with epilepsy. If it is true, then similar networks would be involved in epileptic and non-pathological déjà vu. This was our hypothesis. And what we did is that we compared the brains of the déjà vu declarers and déjà vu non-declarers. What's the difference in their brains? Because we are not able to study the déjà vu as such. We study their brains. And what we studied is, in the beginning, was the morphological differences. So we used a method which was new in the 2010. And this is the source-based morphometry, which is a multivariate neuroimaging technique using structural MRI scans. This is more sensitive to subtle differences in local gray matter volume than voxel-based morphometry, which was, and still it's very widely used for comparing the amount of gray matter between in two groups of subjects. This is based on independent component analysis, and I am not mathematician and no technician, so don't ask me on the details. I am not able to explain. But what we did is that we get the MRI scans from more than 100 healthy volunteers without any neurological and psychiatric conditions. This is important. All the patients underwent the brain MRI and completed the inventory for déjà vu experience. There is one really important question: whether have you ever had the feeling of having experienced a sensation or situation before in exactly the same way when in fact you are experiencing it for the first time? So based on this question, these healthy subjects were divided into two subgroups: déjà vu declarers and déjà vu non-declarers. We had 87 déjà vu subjects and 26 non-déjà vu subjects, and then we used this method, and automatically 8 components were extracted from gray matter segments, and one component then showed a significant effect of group. It means that in these parts of the brain, which are given here in yellow color, there were statistically significant differences in the volume of the gray matter. It was in the basal ganglia, in thalamus. We could see some differences in the mesiotemporal regions, but also in the lateral temporal lobes, and especially in insula. And what's important is the maximum of these changes were found in left-sided mesiotemporal region, a little bit less in the right-sided temporal, mesiotemporal region. And this is another visualization of these differences, which is given here in red. And what's important is that there was a statistically significant negative correlation between the amount of the gray matter in these set of regions. I'm not speaking about the mesiotemporal region or insula separately. We are speaking about the set of brain regions which are somehow involved in this phenomenon. And as you can see, oh sorry, probably it's not working already. I'm sorry for that. But you can see that with more frequent déjà vu phenomena in tested subjects, there was higher differences in their gray matter volume. Angelo Labate and Antonio Gambardella from Italy published some 3 years later a study on the very similar not only subjects but also patients, and they actually evaluated neuroanatomical differences among epileptic and non-epileptic déjà vu, and they had 39 healthy subjects, 22 with déjà vu and 17 without déjà vu, so it's just about 1/3 of our subjects, and they didn't use SBM, but instead of that the voxel-based morphometry, which is less sensitive, but still they found the differences in the gray matter volume in insula. And a little bit later, I guess 2018, some Chinese authors did a very similar study. Again, a smaller amount of subjects, 31 and 67, and again using just the voxel-based morphometry, and they confirmed some changes in the right-sided parahippocampal gyrus with a smaller amount of gray matter in déjà vu declarer when compared to non-declarers. So this is important because this is for the first time that we've got clear-cut statistically significant differences in the morphology of the people who experienced and who didn't these phenomena. This is not so important. I would skip. Also, my postdoc Denshaw then investigated our MRI data using the gray matter covariance, which can be seen as an index of structural connectivity, and the goal was to investigate not only the morphological changes but also the structural, if there are the changes in the structural connectivity between déjà vu declarers and non-declarers. And what he found actually are two networks. These two networks were correlating with the frequency of déjà vu in the healthy subjects, one positively and one negatively. Importantly, both networks were involving these mesiotemporal regions, and using this meta-analytic connectivity modeling then clearly show that there is altered neural signaling within memory-related medial temporal brain structures, which very likely underlies not only the pathological epileptic déjà vu, but also this non-pathological déjà vu. What's important is, and coming back to Walter Penfield and his hypothesis, that there might be some connection between non-pathological and epileptic déjà vu. Frankly, if you have a look on our results, and this is here on the right side, and if you have a look on the voxel-based morphometry comparing patients with temporal lobe epilepsy and healthy subjects, you can see that there are a lot of very similar findings, including this thalamus and basal ganglia, including something on the lateral aspect of the temporal lobe, and also within the mesial temporal region. I'm not sure that it's nicely visible, but if you have a look on the hippocampus, typically in the temporal lobe epilepsy, the maximum of the changes in gray matter volume is in the anterior part of the hippocampus. This is typical for epilepsy. Whilst in déjà vu declarers, all hippocampus is affected, including the posterior part, which is a very nice difference between the temporal lobe epilepsy and the people who are experiencing déjà vu. It's not the same. What's important is also our study published quite recently, which actually compared the changes in different hippocampal subfields. And our hypothesis was that these changes in déjà vu declarers could resemble more temporal lobe epilepsy than schizophrenia. There were altogether four groups of subjects, healthy subjects, which were divided into the declarers and non-declarers, then temporal lobe epilepsy patients and schizophrenia patients. But interestingly, these changes didn't resemble more temporal lobe epilepsy than schizophrenia. So our conclusion was that very likely we are dealing with something like subfield vulnerability to different conditions, I would say. The question is, what does it mean that some parts of the brain, including the hippocampus, are smaller in déjà vu declarers? We know that the hippocampal formation is very plastic. It must be, because of the role of hippocampus in memory. And it is exceptionally vulnerable to the effects of a variety of insults, for seizures, ischemia, inflammation, whatever, as well as environmental and physiological influences. We know that, for example, early life psychosocial stress in animal experiments is resulting in smaller hippocampus of the rats or mice. We also know that the sleep deprivation is reducing the volume of hippocampus, and even prenatal caffeine exposure in one paper was showed that it can reduce the hippocampal volume in animal experiments. So it seems us that these factors, especially when occurring early in the development, have been linked to hippocampal atrophy, alteration of prenatal or postnatal neurogenesis in the dentate gyrus, and neuronal hyperexcitability. This is important because if the hippocampal volume is decreased, in animal experiments, it is linked to the higher excitability of this hippocampus. This is one study published 2011 by Kumar, showing that early life stress and the maternal separation, which is resulting in, which is actually this early life stress, definitely and significantly reduce after discharge threshold in adult rats. And this is just one paper showing that the smaller volume is higher excitability of the hippocampus. This is our study published quite recently, showing that even in a human, some kind of perinatal stress might reduce the hippocampal volume. In this case, this was the mother's anxiety and codependence during the first weeks after birth, and in adults, then more anxiety, smaller volume of hippocampus, and this correlate is significant. This is another study published by my colleague Lenka Lacinova, showing that with more frequent déjà vu, there is increase in the score in the irritability of the limbic system questionnaire, which is to some degree controversial, but still it seems that with more déjà vu experiences, the irritability of the limbic system might be higher. And this is our last paper, not published yet, by Eva Pešlová. In this study, we assessed the so-called fractional amplitude of low-frequency fluctuations in the bold signal. And this is important. This measure is now viewed as the index of brain tissue excitability. There are several papers already published, including epileptic patients, which analyzed the resting state fMRI data, and we analyzed data from 68 healthy volunteers. And what we found is that in different brain regions, which are regions of interest from our previous study, there is statistically significant differences in these fractional amplitude, which probably is supporting our hypothesis of upregulation of cortical excitability in this part of the brain in déjà vu subjects. So to conclude this part of my talk, it seems us that given our data I give you now, the paroxysmal character of déjà vu, the non-pathological déjà vu, and the possible impact of many factors, external and internal, on hippocampal neurogenesis and excitability, it seems us that the role of abnormal hypersignaling, hyperexcitability, and the random internal errors in the brain actually in the genesis of these non-pathological déjà vu experiences is pretty plausible. And now the last part of this my talk, the pathological errors. It is very well known that in the brain, within the brain, we can record these high-frequency oscillations, especially using depth electrodes or invasive EEG. These high-frequency oscillations were for the first time observed in the end of the 20th century. In this moment, they are seen as a promising biomarkers of epileptogenicity. Interictal high-frequency oscillations in different frequencies from 80 to 5 or 600 hertz occur predominantly in the seizure onset zone, which is fantastic. Also, there are papers showing that HFOs are more specific in indicating the seizure onset zone than the interictal spikes. And there is also some papers showing that the removal of interictal HFO generating areas very nicely correlates with good post-surgical seizure outcome when we are speaking about the group data. If, however, we are focusing on the individual data, it's clear-cut that HFOs can be helpful, but not so much as we expected, because the precise localization of the interictal HFO is consistent in only 22% of patients, because there is some movements in the localization of high-frequency oscillations through days. So, and another problem with high-frequency oscillations is that there are not only pathological high-frequency oscillations in the brain, but besides there are also the physiological oscillations. In hippocampus there are quite frequent ripples, which are somehow related to memory consolidation, etc. And also in the fast ripple band there are quite a lot of, probably quite a lot of physiological HFOs. This is a very nice paper published by Birgit Frauscher 3 years ago in Annals of Neurology, showing that these are the actually fast ripples in the this is this fast ripples in the normal brain. You can see that in hippocampus, for example, and in occipital cortex, we can see fast ripples even if these are outside the epileptogenic zone. And in this study, 5% of the channels had a rate of at least one fast ripple every 5 minutes. And these fast ripples were usually below 330 hertz. This is our study evaluating the effect of cognitive task on HFOs, namely on ripples in epileptic hippocampus and in non-epileptic hippocampus. You can see that in both types of hippocampi, we were able to record the ripples, even in non-epileptic hippocampi. And in this study, the cognitive or classical oddball task was decreasing the amount of ripples, but only in the epileptic hippocampus, not in healthy hippocampus. And a very similar situation is with fast ripples. Again, even in the non-epileptic hippocampus, we were able to record some of fast ripples. Of course, less than in epileptic, but still. So the question is whether these traditional HFOs, instead of a biomarker of epileptogenicity, maybe that we can view them as measures of instability of local neuronal population networks, of measures of intrinsic excitability. And it then could be that the high-frequency oscillations of higher frequencies might be more specific for epilepsy. And we had an opportunity to record these high-frequency oscillations with higher sampling frequency. We used 25 and 32 kilohertz frequencies. And we investigated 40 patients with drug-resistant focal epilepsy using depth electrodes. We analyzed 30 minutes resting period, and we focused on the high-frequency oscillations of higher frequencies than the traditional HFOs are, between 500 and 2 kilohertz. And this is a typical example of ripple, of fast ripple. And on the fast ripple, there might be something which is even more rapid, which is very fast and ultra-fast ripple. This is time frequency analysis showing that there might be classical ripples, fast ripples, and then in the higher frequencies, you can see these blobs which are reflecting very fast and ultra-fast ripples. This is not so much surprising that we are able to record these higher frequencies, which are definitely producing in the brain. What's important is that in 95% of our patients, we were able to detect ripples. In 80%, we were able to detect fast ripples, but still in about half of the patients, we were, of these 40 patients, we were able to detect very fast or ultra-fast ripples. And what's important that these very and ultra-fast ripples were observed only in patients with temporal lobe epilepsy, and they were recorded exclusively from mesiotemporal region in our cohort. And even if later we analyzed more patients, still only in one patient there was something at about 1 kilohertz outside the hippocampus, outside the mesiotemporal regions. So this is another examples of very fast ripples, ultra-fast ripples in this time frequency plots. And what's again, it's quite typical is that the higher frequencies can be found in are more spatially restricted. You can see that the ripples can be seen in several electrodes, in several contacts of these electrode in the temporal pole, in the amygdala, hippocampus, anterior, posterior, in the lateral cortex, and in the contralateral hippocampus and amygdala. So ripples can be seen in these specific patients bilaterally, and even a fast ripple we can see in the right-sided hippocampus, but also in the left-sided hippocampus. But these very and ultra-fast ripples can be seen almost exclusively only on one side. So this is one issue, and another is that if we analyze the correlation of the occurrence of these very high frequency oscillations with surgical outcomes, we found that with higher frequencies, the correlation is much stronger. So it seems that interictal very high frequency oscillations appear to be more specific biomarkers for epileptogenic zone when compared to traditional HFOs. But to conclude my talk and back to errors, there's no doubt that like humans, also their brains is not perfect, definitely not. And I'm absolutely sure that also a healthy brain is erring. must be erring. There are many kinds of these errors, many levels and mechanisms of errors within a brain, including these very subtle internal system errors which are not producing any problem in our everyday life, possibly also responsible for illusion, déjà vu. But what's probably more important is these pathological errors in terms of sleeps, which might be something like some epileptic fits, but also the errors of category 2, when there is some bad plan with the behavior of the neurons or glial cells, and maybe that these category 2 errors are resulting in some neurodegenerative diseases. Nobody knows. The question is whether these brain errors are really senseless. We don't know whether it's just an error, just a noise for nothing. And as I said already, if we can, and modeling of the brain is something which is extremely and more utilized. But if we are modeling the brain, there should be also this internal error in the model. We should count with these errors. And that's it. Thank you very much for your attention.
Thank you. This is such an exciting talk. I'm sure you already have a few questions in the chat, but we'll first start with the questions from the
Sure.
audience.
Thank you for a very interesting talk, and now I understand what Mikołaj was saying, that to explore some avenues. This is very
Sorry for that.
very exciting one. So this your studies about comparing the structure of people which experience déjà vu and not, and that you find like differences in hippocampus. I would be like extremely interested if you can find like any other cognitive differences. For example, if they have smaller hippocampus, maybe they have the memory is worse, or some this experiencing déjà vu can predict some other cognitive deficits, or maybe another aspects.
No.
So before you, was it heard? The question was heard on the...
Yeah.
Okay, great.
Yeah. Actually, we didn't do these measurements. Actually, neuropsychological testing was not done in this study. But as far as I know, there are many experiments, psychological experiments with déjà vu declarers and non-declarers done in the past time. And I think that there were no clear-cut differences observed in terms of their memory. On the other hand, it's not so surprising, I would say, because all these changes are not new. This is something which is very likely starting somewhere in the very early in the life. So the plasticity of the brain would probably adapt the functioning of the brain and of the memory. So this is my explanation why there are no clear-cut differences in the memory between declarers and non-declarers. If definitely there are some differences in the hippocampus. But what should be emphasized, I would say, is that these differences are really extremely subtle, and just the statistics is able to disclose them. Yeah.
Another question, but we'll repeat those questions because they are not really well heard.
Okay.
So ask the question, I'll repeat it.
Yeah,
And
okay.
Very nice talk, and I really enjoyed this, the idea of the erring brain. This is kind of a speculative question, but it's interesting to me that the rostral anterior cingulate, you identify this very strong signal of erring brain, and of course there's a lot of interest in that area of the brain as a target for therapeutic stimulation in people with depression. Some of the best results come from apparently stimulating that area of the brain, and I wild thought, but I'm just wondering if somehow the activity in that area of the brain, is that any association with
Depression.
yeah, and the oh, I don't know, sort of complications of thinking, if you like.
So if you can rephrase the question for the audience.
Yeah, Greg mentioned the findings in the rostral anterior cingulate, but it was not during the error. I was speaking. It was error actually. Yeah. So there were some error producing the activity in the rostral anterior cingulate, and our explanation was that it was probably, and the latency was longer than the activation of other parts of the cingulate. So our explanation was that this might be somehow related to the mood and to our emotional evaluation of our mistake. I did mistake, wow. And this emotional part or probably is responsible for the activation of the rostral cingulate. And you are absolutely right. This part of the brain is used for the stimulation in depression patients, in psychiatric patients. So again, it makes sense. The question is whether this electrical stimulation of this part of the brain can help us, for example, to prevent the errors. Probably not. What I showed you is just in this experiment with depth recording and go-no-go task or oddball task in subjects that did the sleeps. What we saw in our experiments is just the reaction of the brain to our error, not the reason for error.
Okay, we've got a question there.
Maybe colleague too.
Thank you for a great talk. I just want to ask a good question about the HFOs that you showed us, because you separated the non-physiological and physiological.
Yeah.
And I just wonder whether they all appear in pairs, whether they are simultaneous, recorded in different brain areas, or they are just separate events. And did you analyze it in this way also?
Yeah, the question was on focusing on the physiological and pathological or epileptic HFOs, and the question was whether these physiological and pathological HFOs are occurring simultaneously or not, and whether we analyze them separately. This is a huge problem actually, because in this moment we are unable to clearly differentiate what HFO is physiological and what HFO is pathological. Unfortunately, the difference is not in their frequency or in their amplitude. Martin Pyle from our research group analyzed these physiological and pathological. Physiological are presumed to be in the non-epileptic hippocampus. So if there are some ripples or maybe fast ripples contralaterally to the epileptic hippocampus, we can assume that this could be physiological. On the other hand, it doesn't mean that the contralateral hippocampus is completely healthy. Actually, in epilepsy is a very strange disease, because it's not just about the epileptic focus. We are dealing with epileptic brain. And because of the disease, we can expect that there are some more extensive changes. We know very well that using voxel-based morphometry, for example, we can see some reduction of the volume not only in the hippocampus and, for example, the thalamus or basal ganglia on the same side, but also in contralateral side. And what's interesting that these voxel-based morphometric changes in the left and the right-sided temporal lobe epilepsy are differing even. So even in the temporal lobe epilepsy, it's not one disease. So it's extremely difficult to differentiate the healthy and pathological, physiological and pathological HFOs. And as far as I know, nobody yet did this differentiation 100%.
The first question which came to my mind after seeing your presentation topic was, like, how do you know that your mind was not playing with you while computing these results? Like, you haven't created any errors in your brain while like justifying the results you created. You know, how objectively say that there is, okay, the science is the science, right?
Yeah.
Can you rephrase it? Because
Yeah. The question, I'm not sure that I understand properly. How can we say that it's really error, for example? When we are speaking about errors,
Yeah, you had some errors during the test, right? You recorded some errors with the patients.
during the test, yeah.
Okay, and then there was a time for your conclusions about these results.
Uh-huh.
And meanwhile, you could create your mind or your team's mind or something was creating errors in the brain.
Yeah. When studying errors, and we can do some more errors, how we can differentiate and what is this issue? I don't know actually. Yeah, definitely. Everybody is doing some mistakes, especially including the research. Yeah, and but still, I think we shall discriminate these errors which are studied in some specific experiments, like this go no go, and our mistakes and errors which are doing, for example, during the analyzing the data. Yeah. On the other hand, the issue is that fortunately, I would say the research in these days is very teamwork. It's very interdisciplinary. There are always a lot of people who are doing the analysis together, who are analyzing the data together, who are discussing the results, the steps of this analysis. So I think that always everybody of the researcher is pushing to do as less errors as possible. And this is something which is extremely important, and I think that this is a problem of whatever kind of research, and of course it can't. This can somehow modify the results, but it's in all the research in all life. So we cannot differentiate, and studying errors doesn't mean that we will not be doing errors.
Right, so we'll take one question from the chat. First question from Daniel. I think it's excellent, but it will be more appropriate for our general discussion. Mm-hmm. So we'll take a question from Konrad Kubicki, and the question is, do you think that pathways essential for unaware errors can potentially play some role in recalling memories or words? For example, slips can be an example of an error in word recalling.
Actually, there are several issues. One issue is whether these subliminally presented stimuli, whether or not they can modify something in the memory. Definitely yes, of course. There is a priming, for example, which is a phenomenon when something is subliminally presented, and then different kinds of stimuli are presented to the subjects who were before presented with this subliminal stimuli, and the time between the stimulus and the identification of the stimulus which was presented subliminally before is much shorter, significantly shorter than other kinds of stimuli. So this is one issue, whether these pathways and these regions which are activated, and we clearly find this activation for the subliminal stimuli within the hippocampal regions, within the mesiotemporal regions. And we know that the subliminal presentation of the words, for example, can go into our memory, even if unconsciously. So this is one aspect. Another aspect was related to some errors, but I'm not sure that I understood.
For example, slips can be an example of an error in word recalling.
Uh-huh. Well, could be, yeah. But, yeah.
Could be. Yeah, the short. I think in general it's about whether these pathways can explain errors in other modalities, like words, for example, not only.
I think that these pathways are more general. it's not just about the very simple. In this task, we presented the letters X and O, which is extremely simple, but still there were letters. Mm-hmm. So probably yes.
Excellent. So it's time that we move to our next speaker. So thank you, Milan.
Thank you.
And please keep writing down the questions. They can be the more general questions we'll leave for our discussion later on. So any questions are good, and all of them are excellent. So now we're going to switch gears a little bit, and we're going to move to the next presenter. And we're going to probably ask for some changing the slides. Okay, great. We've got it here. Do you have that pointer? Oh, me. Okay, so Professor Gregory Worrell from Mayo Clinic in the United States. I did my postdoctoral research in Greg's lab, and it was really career-changing for me to have such a mentor who gave so much freedom again to a young student who didn't even have a PhD title yet, because I was about to, I submitted my thesis and I was about to get it defended, but still Greg gave me a lot of freedom in doing the research. And in staying in this lab was really like mind-opening, just like the talk that you've heard from Milan, that you can even investigate things like that, questions like that. That was for me also equally mind-opening that to come into Greg's lab. And the technology that I got to learn and the questions that we got to answer in the end of my postdoctoral research were, I didn't think that we could be answering those questions when I started. Like to stimulate the brain electrically and to get enhanced performance in psychological functions like memory was like beyond our imaginations at that point. And yet, Greg is an example of another person who has got this vision, I think, who's characterized by a vision, that he's got vision and courage to dare to ask questions which are ahead of people at the time right now. And the technologies that you're going to hear today about, they are really like from a science fiction movie. So I really encourage you to like open your mind to, what you'll be hearing about, what it means for where we're going in the future, and what it means for us, both clinically and kind of ethically or philosophically, where it's all heading and what's possible right now and what will be possible in 10 or 20 years or so. So Greg is going to be talking about, some interesting devices, also in epilepsy patients. But I won't tell anything more. I'll just give the ground to Greg and welcome you. Thank you.
Thank you for the very nice introduction and fantastic opportunity to see old friends and meet new friends. It's really an honor to be here, and you're the Gdansk University of Technology, spectacular place, beautiful city. We've had a couple of nice nights out, and it's really been a wonderful visit. So, yes, thanks for having me. So I'm going to talk about devices and a primary idea, which is it's not that complicated, and it's partly driven by the technological limitations of where we are currently, but also coupled with the technological opportunities that exist currently. Because I'm very interested in getting therapies to patients, well, in 20 years. Sure, I'm interested in that, but I'm really interested in doing that now. And so a lot of what I talk about, you'll see that as a theme. And one is this idea of integrating brain implants with local and distributed computing, and I'll tell you more about what that means. It's a very epilepsy-centric talk, but I think epilepsy, just as Milan showed, the opportunity to work with our patients and it really is a platform disease to learn about the brain and create new technologies to help patients. And and that's not a new phenomena. That's actually throughout the history of neuroscience, the disease of epilepsy has really played a powerful role. How does this advance? Okay, there. Oh, maybe I had the wrong button. Yeah. So just a couple of disclosures. I do have licensed some of these ideas and technology to accompany Cadence Neuroscience. Some of the device work that I'll tell you about is with the company Medtronic, which is obviously a large medical device company. But most of the work, well, really all of the work has been funded through the Brain Initiative, Epilepsy Foundation, DARPA, and really grateful patients at Mayo Clinic, as well as the ICRC, which is a Czech entity, and how I met Milan and his team. This is a little bit of an older figure from the lab, and you'll recognize that guy in the back there. So this is probably from about, I don't know, 5 years ago or so, but Milan came to the lab and really pushed us forward in the area of cognition, which I haven't done a lot with. It's been a fantastic voyage as a bystander, seeing it. I see it in my patients all the time. It's a very common comorbidity of epilepsy, verbal memory deficit. It's rare to see an epilepsy patient that doesn't have a complaint, a cognitive complaint. Now, they also have complaints about sleep. They also have problems with mood. So it's much more than just the seizures. So I'll tell you a little bit about the history, not much. The current state of where we are with brain stimulation in epilepsy, responsive neurostimulation, and deep brain stimulation, and where I see the gaps currently in those technologies, where the opportunities are. And I think we can sum that up fairly, to simplify things, I like to simplify things, is where to stimulate, when to stimulate, how to stimulate. And then we have to be able to, and this is a huge piece, which is, one of those things that it's just engineering, but it's not been accomplished. We need to be able to actually track the outcomes. So seizures are really tricky things. They don't leave behind any sequela. They don't produce a signal in the brain after they've occurred. So it's not like tracking someone with, say, a brain cancer that grows and you can see if it grows or shrinks. You track the effect of the therapy with patients. Right now we do that by them telling us about these events that have happened at a different time. And we know these events are often, as Milan was pointing out, amnestic events, subtle events. So it's a very impoverished set of data that we work with. So to me, that's just engineering. That's not a hard thing. We can record from the brain. We should have a better way to track these outcomes. And that really drives one piece of this idea of next generation of devices. And so just in a general talk like this, I always like to just say a little bit about epilepsy, because has been throughout the time of humankind misunderstood. It's very common. It's about 65 million people worldwide. And even those in first world countries that have access to drugs, about a third continue to have seizures despite taking medicines twice a day, every day. And as I pointed out previously, it's much more than just the seizures. In fact, some of these, what we think of as comorbidities, or side effects, if you will, of this disease, are as disruptive as the seizures themselves. Now, on the good side, we have a lot of drugs. We even have an excellent device, a low glycemic diet or ketogenic diet, epilepsy surgery can be very effective, and what I'll talk about, devices. I just want to say, epilepsy surgery, so that's the surgical cavity from an anterior temporal lobectomy. Again, sometimes there's a misconception that this is a very small little surgical strike. This is usually a fairly large area of resection of brain. So what about electrical stimulation? So again, 30% roughly, we would say, of these 60 million people would continue to have seizures despite medication. So how could we use electrical stimulation? And this is not a new idea at all. In fact, probably the first use of a therapeutic use of a device with electrical stimulation is back about 1950 by this guy Heath, who had patients with externalized electrodes and was delivering stimulation. But beyond that, multiple areas in the brain have been targeted. This is a target for one of the first implanted devices back in the early 70s, the target, the cerebellum. And, you know, there's evidence for some effectiveness in multiple sites, and also probably the most common site stimulated currently, it's not brain stimulation, it's actually vagus nerve stimulation. But out of all of these studies, this is a little bit US-centric, of course, but what's FDA approved in the US is this responsive neurostimulation. I'll tell you more about that. And then stimulation of the anterior nucleus of the thalamus for epilepsy. And this is a little bit older data. I'll give you the update, but about 66% reduction in seizures, and about 60% of patients were responders. These are from the Class 1 evidence trials. Now, I want to belabor a little bit just how these were studied, because I think this is one of the gaps. So those very well-designed trials, prospective, randomized, double-blind, patients are implanted with the device. It's sub-threshold stimulation, so they don't know whether they're being stimulated or not, and you're tracking a diary. So you're able to do a very well-controlled study where half are getting stimulation, half are not. The physician doesn't know, the patient doesn't know. So so you keep a diary, device implanted, and then randomized to stimulation on or stimulation off. And these results were really quite remarkable, because there was a significant reduction even in those that didn't have the device turned on. Most of that's likely a lesional effect, but the other thing that we know, and I'll show you data for that, again, you're counting on diaries. Patients are very poor reporters of seizure events. We know that. So this isn't ancient history, this is fairly recent, 2003, where were we? At that time, if you had a patient like this that had drug-resistant epilepsy, you were evaluating them for epilepsy surgery. This is a gentleman, about 20 seizures per day, and normal MRI, so he's implanted with a large convexity grid, 64 contacts and 8x8, and recorded where his seizure came from, and a nice focal onset from this single contact here. So you see it in the adjacent contact too, those two adjacent contacts. So just perfectly what you want to see. But unfortunately, you know where this is going, that's motor hand. So not a surgical candidate. So even as recent as 2003, a patient would have gone through this procedure, a highly invasive diagnostic procedure, and not actually had a therapy at the end of it. Would have been determined to not be a surgical candidate. This was our first patient that we implanted with the NeuroPace RNS device. And so that's a device, it's really a sophisticated piece of engineering, quite in advance.
a microprocessor in the device. You can see it connects to 2 4-contact strip electrodes. There's actually 3 strips, but only 2 of them are active. So 8 electrodes. You can record activity, sensing, that was the big advance, and then you can actually program a detector on the device and actually stimulate. And that's what's shown here, which is no small thing at that time, and even still currently, what you see is this is 1 second from here to here, and between these hash marks. And you see the onset of a clinical electrographic seizure here. The device detects it, delivers a stimulation, and actually aborts the seizure. So that's a patient out living their life, taking their medicines, going about their activities, and a device that's actually able to detect the seizure and deliver a stimulation. So very powerful proof of principle. Now it turns out that it's very difficult to stop a seizure once they start. And one of the things that we've worked on for many years now is how could you make this better? I mean, you can see the limitations. You're recording from 4 channels. You've got 8 electrodes. Detecting these events, we know from microelectrode recordings and optical imaging, by the time you record a local field potential that you see on one of these macroscopic electrodes, there's millions of neurons involved. So that snowball is rolling down the hill. It's very hard. It's very hard to stop. So the ideal, could you forecast seizures? Could you identify periods of increased seizure risk? Is something of considerable interest and potential. The other big device trial was the Sante, the stimulation of the anterior nucleus of the thalamus. That was actually, the trial was done earlier, but it was approved later than NeuroPace. But that's 2 leads in the anterior nucleus of the thalamus. That's approved really throughout the world. It's approved in the US, it's approved in Europe with the CE mark, it's approved in Australia, in Canada. So very widely used, and that technology stimulates the anterior nucleus of the thalamus to prevent seizures. Approved in 2018. I told you about the NeuroPace technology, which was approved in 2013. And now we have, I told you I would update you on where we are with these. We now, which is very nice, we have long-term data from these devices. Dr. Salanova and Dilip Nair, Salanova for the Sante, stimulation of anterior nucleus of the thalamus. And what's been found over time is these therapies get more effective, which is nice. Now after over seven years, 75% reduction in seizures, about 70% responder rate. Milan and I were talking about this just today. But it remains rare to get a patient seizure-free. So all this technology, all this work, but rare to have a patient seizure-free. And just this caveat here, this is a fantastic number, 18, because these are the most difficult patients, of course. 18%, let's just say 20% have a 6-month seizure-free period. What that means is over the 7 years that this study has been tracked, this patient will have a 6-month period where they're seizure-free. And then they have recurrence of their seizures typically. Very similar results for the responsive neurostimulation. Improvement in quality of life. So really impactful for patients, but again, rare for someone to be seizure-free. So that to help you understand what that means, these are paroxysmal events that are seemingly random where people lose consciousness and they may convulse or become unaware of their surroundings. So that happens randomly, maybe, once a month, twice a month, or once every 6 months. it remains incredibly disabling for patients. So what have we learned? That is where we, let's say 5 are currently, they're palliative therapies. These are not rendering patients seizure-free. Therapy optimization is slow. To get there, to get those results I showed you, which are very impressive, that's over the course of 5, 7, 10 years. So how do you accelerate that therapy effectiveness? That's not just after putting the device in immediately. Really, what we need is a biomarker. We heard about one that Milan was talking about earlier, HFO or interictal spikes. Seizures are these, if they occur once a month, that's a relatively rare event. And if you're adjusting your therapy based on those relatively rare events, it takes a long time to make decisions and adjust therapy. Comorbidities, I mentioned. So none of this technology that I've mentioned so far is directly targeted at the comorbidities. And the very common comorbidities, as we've talked about, are memory, mood, and sleep. It's almost without exception that a person with drug-resistant epilepsy wouldn't have issues in all three of these domains, in addition to their seizures. And then something that's really been laid bare during COVID is how patients are currently cared for, this idea of the point of care. So with devices, or similar with drugs, if you're going to change drug levels, but with devices, patients generally have to come back and see you. Well, during COVID, that was a real problem. Now, clearly the technology, this is an engineering problem and a regulatory problem, but an engineering problem. Clearly, with the Internet of Things, couldn't you control those devices from a distance? So this idea of the point of care model is really difficult for devices, and I think an area where we can make a lot of progress. I'd mentioned the diaries. That's another big piece for epilepsy, but that's also true for psychiatric diseases like depression. Decisions are made. A patient has this disease. They live with this disease. Then they come and see their physician. And in that 30-minute appointment, if they're lucky, 15-minute appointment, they are asked, how are you doing? Well, it's hard to describe how you'll tell you how you're doing right now, but you really will not learn much about how they've been doing for the 6 months since they saw you last. So the idea of the point of care model, this will be overturned by the connection with technology, I think, and deliver more effective therapies. So with epilepsy, again, as if you will, a platform for moving these technologies forward. Could you forecast seizures, deliver stimulation, change stimulation therapy, not while the patient's in your office, but while they're living at home? Could you make adjustments in their therapy to help their mood? Could you change therapy to help their memory? Could you change therapy stimulation to help their sleep? Well, to do any of those things, you would first have to know how they're doing. You would rather them not come back to see you every day to ask those questions. So you need to do this in a more technologically enabled way. All of that's possible. So we need new devices, we need analytics, and that's my long-winded way leading us to the second part of this talk. So now one is, so we're going to need to record from the brain, because we want to know, because Milan's already showed us that we can record from the brain and detect how the brain makes errors, or at least track where those errors come from, and maybe you could stimulate to improve those errors. You first got to record. You first have to have a biomarker of that phenomena. So this question of how best to record from the brain, it really is a I think, in terms of application for disease, and also true for understanding basic physiology, it depends on the question. It depends on what you're trying to look at. But of course, you can record across a wide dynamic range, spatially or spectrally, the frequency of activity. And the brain, you'll appreciate, there's a lot of engineering legacy on how devices and how technology has been used to record from the brain. So up until 15 years ago, which as you get older, 15 years doesn't seem like that long, but it wasn't that long ago, that the dynamic range of what was largely paid attention to was this range from, say, 0.1 hertz out to no more than 100 hertz. And that's probably being generous. And as we heard from Dr. Brazdil's talk, the brain generates much more of it than that. Of course, the brain, now we haven't talked at all about infraslow activity or DC, but the brain generates electrical signals from DC changes to ultra slow changes that occur over a day, all the way out to very fast activities. And of course, as Dr. Brazdil was telling us, even out beyond 1,000 hertz. I need to update my slide, because I'm still at 1,000 hertz here. And this is for local field potentials. And of course, it depends on how you measure from the brain. Are you doing that with these macro electrodes, like what I'd shown you in that patient, recorded that seizure, and most of what Milan had shown you, or from, say, a 40 micron wire where you can record single units. The spectrum of activity that you record from those technologies is very different. And very interesting. So it makes it, you can do PhD theses and a lifetime of activity. I showed you a seizure earlier that looks something like this, and this is a 2-second time scale here. And this is a depth electrode, like Milan described. So these are these macro contacts, how we clinically record from seizures. And then also, in addition to the macros, these are 40 micron wires here, and these are the traces here. Now you'll see they have less activity. They're locally referenced, so you only see the very local activity. And then these are the macro contacts, these bigger contacts, 1 through 4. And then there's additional micro wires embedded in here. And this is our classic seizure, this buildup of rhythmic activity. It's about probably 20 hertz oscillation. Clearly a seizure, clearly different than the background. But if you blow this up, and this is relevant for this idea, say, of neuropace, responsive stimulation, that you want to detect a seizure and then deliver a stimulation to stop one. What you see here is when I blow up this region, well, on that micro wire, that's before this very clear local field potential oscillation occurs. This seizure starts at about 360 hertz. So you're not going to record that unless you're sampling at about 1,000 hertz. The clinical device is, the NeuroPace device samples at 200 hertz. And on the macro electrode, you can also pick up a fairly high frequency oscillation. And you can record higher frequency oscillations on these clinical macros as well, but just with this example. But so clearly here you can detect this seizure earliest if you're recording at the level of the microdomain. So, and seizures probably are phenomena, I'll go out on a limb here, have never really proven this, but we've written about it. Seizures are phenomena that probably start on the scale of a cortical column. They don't start at the level of a single neuron, but they probably start at the level of the organization of cortical columns. And to sample that level of organization, ensembles at that spatial scale, you need to you need microwires. And of course, there's lots of brain that you're not sampling from. So it's a very complicated problem. But electrophysiology is important. We all love electrophysiology. Now, what about at the single neuron level? So what happens in seizures at the single neuron level? And this, I'll just tell you about some work that Mark Bauer did in the lab when he was a postdoc with this. So this is, again, these same electrodes and microelectrodes protruding from the tip, and then there are microelectrodes in the shaft. And I know Dr. Krusowicz is interested in using these kinds of electrodes. And the fact is that you can get very nice unit activity from the shaft electrodes and from the tip. And you can record that over long time scales. So these are patients implanted for what I call prolonged monitoring. So maybe if you're doing it in the OR, that's acute. Prolonged in epilepsy, if you want to record seizures, you're not going to capture those in the OR, right? So you're going to have to implant the patient, keep them in the hospital maybe for 5 days, 7 days, sometimes even longer, unfortunately. There ought to be a better way to do it, but we won't cover that in this talk. But you may record for 7 days, and the fact is that you can hold many of these units. You'll lose many of them, but you can hold them over long time periods. So this is a patient where we recorded over multiple days, and this just shows the 45 minutes leading up to a seizure. And this is in the hippocampus. The seizure begins in the hippocampus. I don't know, so what do you think happened? So I always imagined you'd see increased neuronal firing, might slowly build up or quickly build up. That's really the paradigm of seizures that we've always had. So I love this result because it still puzzles me. So this was 7 patients. It was about 10 terabytes of data. So this is a technology institute. So 10 terabytes of data, and when this was like 2010 or so, just managing that data, streaming that data out of the patient's room, running automated detectors on that data is a bit of technology, but that's certainly all it is. It's just engineering. It's all doable. And these are what these microwires look like. They're about the size of a human hair coming out of the tip. So those go into the brain. If you use them in the medial temporal lobe, into the amygdala, into the hippocampus. And we had 200 microwires. And so as we looked at the seizures and multi-unit activity, what we saw was that about 8% of the units increased prior to the seizure. So, yeah, that's nice. That's what should happen. But actually, about 9% of them decreased. And again, I'm talking about a seizure that is recorded between these 2 electrodes that are 0.5 centimeters apart. So 5 millimeter apart, recording a seizure, and the units that you can pick up right around that. So 8% of them increase, 9% of them decrease. And the thing that still is remarkable to me, most of them don't do anything. The brain is not a bag of saline. So it's highly compartmentalized, and this, I think, speaks to how the brain is organized to do what it does. So neurons that are right next to a seizure focus, and these are very large amplitude, 100 millivolt signals, that neuron just keeps firing away just like it had, for the last 12 hours. So it's compartmentalized at the scale of, well, certainly submillimeter, but it's compartmentalized at the scale of microns. So the extracellular space is very complex And what it does is create an environment through which single neurons can fire and not be disrupted by the neurons right next to them. So this was always, again, still surprises me. Now the next thing that Mark looked at, what about the neurons that fire together at the onset of the seizure or early in the seizure? So we know from this idea from Donald Hebb, neurons that fire together wire together, and we think of that as that's a learning mechanism. So you repeat a task over and over and you learn. Well, there's probably no stronger task or no stronger activation in the brain than a seizure. So Mark's idea was that should really activate this phenomena of Hebbian learning. Neurons that fire together at the onset of the seizure, that should be a learning mechanism. That's a very interesting idea. That suggests that you learn epilepsy. There's some evidence for that. As you have seizures, it goes all the way back to Hewlings-Jackson, who said seizures begot seizures. So it wouldn't be surprising that repeating seizures over and over strengthens those neural connections. So what Mark did was look at what happens, neurons that fire together prior to the seizure or just at the onset of the seizure, and then looked at what happened in slow wave sleep after seizures versus slow wave sleep in the background or just in general. And what he found was that neurons that fire together at the onset of seizures recapitulate that during slow wave sleep. And that's a phenomena that's believed to underlie learning and memory, this idea of consolidation. It's a concept that has really deep molecular investigations, which ours wasn't. This is just recording from a handful of neurons that we're able to record from, but it's an interesting idea, and it has an interesting therapeutic idea. Well, if most of the learning in seizures happens in slow wave sleep after they have the seizure, wow, that might be when we should intervene, because otherwise the human brain's, when that hippocampus is not seizing, it's actually doing that's something that we haven't talked about. When it's not seizing, it's doing normal things. And that's the challenge with epilepsy surgery. You're not just removing a piece of tissue that doesn't do anything. When it's not seizing, when it's not creating problems, it's encoding memories. If it's hippocampus. So it's this idea that you might want to stimulate the brain depending on brain state. Is that possible? Well, you'd have to be able to, one, record from the brain. You'd have to know when seizures occur, and you'd have to be able to stage sleep, stage behavior with electrophysiology. So that's that. I've told you about we want to track seizures. Now I'm telling you we want to stage sleep because we might want to stimulate differently depending on behavioral state. And then this lofty idea, what if we could forecast seizures before they occurred? And this is a schematic from an important study that was published in 2013 by, I call them the Australian triad, Mark Cook, Terry O'Brien, and Sam Berkovic, using this device created by NeuroVista. And the NeuroVista device implanted recording device in the subclavicular area and then 16 channels of EEG. And what they were able to show was that there are signals in the local field potential that show changes even up to hours prior to seizures occurring. So that seizures aren't completely random events. Now, the other thing that came out of this study that wasn't quite so exciting as seizure forecasting, but Milan's seen this figure a million times. We all have in the epilepsy world, because it was the fact that if you looked at patients only 5 of the 11 patients that were keeping these diaries were accurate at all. So most patients, again, because these are amnestic events, maybe they occur during sleep, they go unwitnessed, patients are not good at reporting their seizures. So think of yourself, if you're a physician in the office, your patient's coming to see you and they say, no, I haven't had any seizures. I'm doing just fine on the medicines I have. Well, maybe, maybe not, because often that data is not good. Or it can work conversely. Oh, doc, I've been having lots of seizures. And we all know, you really just, it's very hard to know. It's very hard to know. So that's 2 important things that came out of that study. Another important thing that came out of that study and the NeuroPace study was when people started looking at this long-term data. This is from the NeuroPace device study. Now you're collecting seizures over months and months and months. So you have data. Just like the NeuroVista study, you have data. So you can start asking, do seizures occur completely randomly? And this is from Maxime Baud, who showed that in patients, this has been known for a long time, that seizures tend to have periodicities. Patients have their seizures, say, around 7 or 8 o'clock, or they always occur on awakening. So are these periodicities, patterns that you could use. But what they showed that was also very interesting was that there are these longer time scale periodicities that probably patients aren't so much aware of, because the temporal scale over which they occur, it's just hard to wrap your mind around that. But that many patients, on average, would be more likely to have seizures with this periodicity of 20 days. So that's what we know. So what do we need? Well, this is the idea of integrating an implant with local and distributed computing. So first, you can't put all this technology in an implant. Yeah, what moves really commercial electronics moves really fast. What moves really slow? Medical electronics, especially implantables, because there's all this regulatory. It's important that it's there, but it just makes things move slow. So the idea that if you can get the data seamlessly off the implant onto a handheld, think how fast the capability of your iPhone has changed. So if you can get the data off the device in a seamless way onto a handheld platform, and then of course you're coupled to the cloud, it's infinite computing basically. And now you're moving at the speed of commercial electronics, not the speed of medical electronics. I'm making this sound easier than it is. Of course, there's still all the regulatory problems and such, but it's possible. And so this was the device that was available. It was an investigational device created by Medtronic that was rechargeable. And that was one key piece, because if you're going to telemeter data off a device, that takes a lot of energy. So you need to have a rechargeable cell. So it's rechargeable. It's 16 channels, 16 contacts. You can record from any 4 channels. So if it's a bipolar, any 8 electrodes across those 16. And you can change that dynamically. And it has a bidirectional interface. So you can write algorithms on a handheld to measure things and then change therapy. You can program. It has some processing on the device, but not much. Not much. So you can do some of, short latency detections, like denoted here, that would be like the RNS, so you could detect an event and deliver stimulation on it. So if you need really fast latency, you need simple algorithms, because those algorithms got to live on the device. But since you have these more sophisticated algorithms, you can run those off the device. And what we created here, this is all part of a Brain Initiative program, was something we call the epilepsy patient assistant device. And there's a large team of people that have worked on this, led by Ben Brinkman, Taupal Altia, Mona Nassari, Vladimir Sladky, Philip Milvat, and I'm sure I'm forgetting some, so forgive me. But so there's a big engineering team behind this handheld that can do a lot of things. It can also enable a way for the patient to act with the device and synchronize those signals. So say a seizure can be detected, and then the device can actually, the handheld device can ask the patient what they're feeling, can give the patient a memory phrase even. So it enables this bidirectional communication between patients and devices. And then you can run longer time scale, latency of events. If you can actually predict a seizure, forecast a seizure an hour before the event, well, you don't have to intervene within milliseconds. Those are really long time scale changes that you might be able to make to reduce the excitability of the brain. So that was the idea. So where do we do this first? Do you just put it into a human and say, sure, hope all that works? Sometimes that happens in medicine. It turns out everything I told you about human epilepsy, just insert the word canine. Turns out epilepsy very common in dogs. And dogs are large enough to accommodate human-sized devices. So it's not like trying to put a human-sized device in a mouse or a rat. It's not going to work. So dogs have actually played a big role in testing anti-seizure medicines, and we've done a lot of work with devices. It turns out this is a pet dog. So this isn't a research dog. This isn't a dog that someone gave epilepsy to in the lab by a traumatic brain injury. This was a dog owned by dog, and everyone knows how dog owners are about, I have dogs, know about how people are about their dogs. So there's been a lot of interest in how do you improve therapies for canines as well. So this is a dog with epilepsy. This is the epilepsy surgeon, Beth Sturgis, epilepsy EEG technician, Chelsea Crow, at UC Davis, where this dog was implanted. So it's a pet dog with epilepsy implanted with this next generation device. And I like to tell this story. People ask, well, how do we know this is going to work? Stimulating anterior nucleus of the thalamus. I mean, this is my dog. Before you put electrodes in my dog, how do you know this is going to work? Well, my answer in that case was, well, we've tried it in about 20,000 humans, and it seems to be working pretty well. So it marriages the idea that you have an FDA-approved, very well-studied technology. Now you're just adding what my friend Tim Dennison, who was formerly at Medtronic and now at Oxford, calls the scientific payload. So you have an approved therapy, and what you're adding is a scientific payload. So we're going to add sensing and streaming to this FDA-approved therapy. We're going to do it first in dogs. And so that's what this shows.
Hold it for a minute.
This pet dog, he's got now this is made a little more complicated because he's doing a navigation experiment. He's got a GoPro camera on his back, very similar to sort of navigation that we're doing in humans. And he's got this nice beta alpha rhythm as he navigates. People that know about rodent navigation recognize that as a good rhythm. Also, of course, demonstrating that dogs are highly motivated by food, just like humans. So how are we going to track these canines with epilepsy or humans out in the field? So we're going to stream this data off the implant onto a handheld. Of course, the dog's not going to hold that. They're going to have a vest. And then it's going to stream either through a cellular service or through Wi-Fi up to the cloud. Then you're going to have all this data that you're going to want to manage. And this is a dashboard that a team of engineers led by Václav Kremen created. And And we're going to track things like the battery. We got to recharge this thing. If the patient, so it does have patient burdens or pet owner burdens. They've got to charge this device. What this shows is, of course, over about 20 days, the device slowly starts running down to about 50%, then they charge it, down to 50%, then they charge it. The data rate, this is actually pretty good. It looks like a lot of data is being dropped, but that's how Bluetooth telemetry works. So you got to have a lot of error checks and a lot of technology, I guess, is, from the point of at the end of the day. And then you can track interictal spikes. You can track seizures. And for the first, I shouldn't say, I always hesitate to say for the first time, but you're now able, while a patient's outliving their life or the canine's outliving its life with the owner, to have an automated detector to detect seizures and create labels on the data. And you make that detector hypersensitive because you want to detect everything, and then you have the experts go back and annotate that data and continue to fine-tune your machine learning algorithm that's going to detect and manage all these seizures. And so you can, at a glance, then look at data. You can pick your subject M1. You can see how much data's been streamed, how much was captured data over the last 24 hours, how the battery's doing, how many seizures they had. You can track when those seizures occur. So this patient typically has seizures around 9 o'clock. They either have seizures in the early morning when they wake up or around 9 in the evening. That's pretty common for temporal lobe epilepsy. That's an important piece of data. Seizures aren't totally normal, or totally randomly distributed. So you can probably use that. And as you collect this kind of data, you can adjust how you do the stimulation. And this then just shows what the stimulation, and what the expert does is just go and mark, this was detected by an algorithm, and then just goes and marks the onset of that and the offset. And then again, you can use that in as that labeled data to fine-tune your detectors. So this is what it looks like. And if you track seizures over many hours and over many days, in this particular canine, what happens is they have clusters of seizures. So the best, in this animal, the best predictor of a future seizure is them having the first seizure. This is pretty common for, certainly more common in pediatric childhood epilepsies, but, and I'll say, parents don't need all this technology to know that. They know if their kid has a seizure, there's a good chance that they may have one in the next hour, and you can give them rescue medications. But that's not always the case. So, but this dog clearly has seizures that occur in clusters. But now what you really want to predict are these really longer time scale events. You had a cluster here, and then about 30 days later had another cluster. Can you predict that? And this is work that Nick Gregg has done in a population of dogs, and shown that, just like in the human work that I showed you from both the NeuroVista trial that Pip Caroli analyzed and the RNS trial that Maxime Baud analyzed, is that seizures occur not randomly. They occur at particular times of the day. I don't have that here. Particular days of the week and particular months of the year. So they're really this very complex and not completely random distribution of seizures. Now, this is, again, as I warned, this is a very epilepsy-centric talk, because that's what I do. But people who have seasonal mood disorders, people who have depression, will tell you very similar things, that there are often these cycles. So it's not only in epilepsy. This is something common, I think, in general to the in general to the brain, and many diseases have not only circadian but multi-day rhythms. Here we want to use that to better treat epilepsy, to adjust therapies. All those patients historically that have, or most that have these periodicities, they receive a fixed program of stimulation, and they take their medicines, I'm just using an example, at 8 in the morning and 7 p.m. And that's what they do. And that's very helpful, because if they didn't have that, they would have more seizures. But when you see this kind of data, you really ask, wow. And it's called chronotherapy. It's not a new idea. Can you more intelligently deliver therapy? Whether it's a medication, whether it's a behavioral therapy, whether it's a brain stimulation. And we think the answer is yes. Now I'll disappoint you because I'm not going to have data to prove that. Just that's a forewarning. I'm telling you everything I know, which is not enough, okay? But we don't have that. This is just, this was Vladimir Sladky and Peter Nedley. You will notice a lot of Czech names. I have this nice collaboration with the Milan's Hospital at St. Anne's, and we have, over the years, many Czech researchers. But this is precision recall for, these are NeuroVista humans. This is RC plus S. UCD 1 and 2 are pet dogs with epilepsy in California. All that data is coming up to the lab, and we manage that, detect seizures, and have looked a lot at forecasting. This is even some older data, but it turns out you can forecast with, and they're probably not going to surprise you that you can forecast after what I've shown you, that these seizures occur in circadian patterns and weekly patterns and monthly patterns. You could, come up with a hazard model that as you get closer to Thursday, you're going to want the probability to go up. And what this does is just compare that performance of forecasting with a random predictor. So you can do better than a random predictor. Not so surprising, but wasn't thought to be true a decade ago. Another piece that's really important here is the time, and there are many days where there are no warnings. So rather than taking a medicine twice a day, every day, for events maybe that happen once a month, maybe there's some days you don't have to take medicines. Some days that you don't need brain stimulation. There's certainly days when you're going to be at increased risk of having seizures. So all of those are possibilities. That's a picture, I think, of Ben Brinkman's dog that got on the cover of Brain. So that's, your dog can get famous, right? So that was the dog work, and then we got approval from FDA. I told you there's a big regulatory piece of this, but the regulatory piece is made easier by the fact that we already have an approved anterior nucleus of the thalamus stimulation. So what we did, one of the advances with this device, it's a 4-lead device. So you can implant 4 targets. I think in the future we'll probably see even extensions where you can implant additional nodes that maybe rostral anterior cingulate for depression. Depression is very common in epilepsy. So you have FDA-approved reasons for putting electrodes in the brain to treat the epilepsy. Can you extend that concept of a scientific payload, or add? It certainly adds risk, but that's how medicine moves forward, and it has to be acceptable risk. But so this is a 4-lead device. Electrodes in the bilateral anterior nucleus of the thalamus and bilateral hippocampus. So both of those are FDA-approved targets for electrical stimulation. And this is just some of the FDA speak. Obviously, things have to be tested on the bench. They're tested in animals. They're tested and retested before they're implanted in humans. And we now have this device implanted in 4 patients. And I'll just show you a little bit of that data. Not surprising, these are the 4 humans and the 2 pet dogs. So those are people and dogs out living their lives with their owners in their home, data streaming up to the cloud. Very good automated seizure detection. These are pretty big signals, and if you're not trying to do very short latency detecting the exact onset, not so hard to detect. That's not a small deal. You now know when you see that patient in the office, or I know actually when they're still at home and I log onto the computer while I'm in Czech Republic, or while I'm in Poland, or while I'm on vacation, I know how many seizures a patient has because it's all captured there. Not only do I know it when I'm in the office. So this really opens an avenue. I think this has been a real gap for epilepsy care, just being able to quantify the primary outcome, the number of seizures. I mentioned we wanted to track brain state, so can you do that? So that's a really well-developed area of neuroscience, recording from the scalp to detect what state of the brain. So are you awake? Are you in REM, rapid eye movement sleep, or in slow wave sleep? And these are just some representative examples from the scalp, and below are signals from our patients today. So we have simultaneous scalp somnograms and streaming intracranial EEG. And then we use this as the gold standard, which the scalp is, the gold standard to stage. And the question is, how well can we stage sleep, stage behavioral state, with intracranial recordings? And the answer is it goes pretty well. This is automated hypnogram created from recordings from bilateral anterior nucleus of the thalamus and hippocampus. And remember, why we wanted to do this is because we know sleep's it's a common comorbidity. People with epilepsy have fragmented sleep. We raised that interesting question, would you want to stimulate in post-ictal slow wave sleep to disrupt consolidation? Could you stimulate during slow wave sleep and enhance consolidation of normal memories? These things have been tried. So the performance is pretty good. It's complicated by the fact that we're doing brain stimulation. So here, this is all anterior nucleus of the thalamus. So no electrical brain stimulation, 2 Hertz brain stimulation, 7 Hertz brain stimulation, anterior nucleus of the thalamus stimulation, and then high frequency, which is what the Santi results are. And the performance is pretty good. It becomes more challenging, and this won't surprise you, to differentiate REM from wakefulness with just intracranial. So that, you can see these are our area under ROC curves, so you can see that can be a little more complicated. But overall, the performance is quite good. You can actually manipulate sleep. Probably not surprising that you could turn stimulation up and wake people up. Yeah, well, that's not so exciting. But these are really early days. Questions are, can you better? The patient I showed you, they have seizures at around 10 a.m. or 9 p.m. They don't have seizures at night during sleep. And I know that not because they tell me, but because we're recording continuously. It turns out actually when they're in their home sleeping, that's the best data collection we get because the telemetry is very good, the data drops are lower. So as long as they charge the technology, which is 3 devices, there's a big patient burden. We need to make this better. But so the idea that could you actually improve sleep, we're working on that. We do have, this is from Nick Gregg, just recently submitted paper that with stimulation, we can actually change that not the circadian rhythm, but that longer term rhythm. So these are interictal spike rates. over the course of many weeks, 25 weeks. And then this is a moving average in the orange. And then the red dots are seizures. And what you see is there's this longer time scale periodicity in interictal spikes that does correlate with seizures as well. It's certainly not one-to-one, but this interictal spike rate. And what Nick has shown is by increasing this should actually be current, increasing the current, you can have voltage clamp devices or current clamp devices. The new devices are current clamp. But by changing the stimulation current in the anterior nucleus of the thalamus, he was able to change this multidian, or this higher multi-day cycle in spikes. So it went from 11 days down to 8 days. So at least showing that we can use electrical stimulation. One, we know we can reduce seizures, but we can also manipulate I presume the excitability of the brain, here using interictal spikes as a biomarker of excitability. What about verbal memory? So yeah, you see the patient in the office, they say my memory's great, or they say it's bad, or I don't know, they say they don't know. Well, now with this interface that we have, we can record electrophysiology, we know how we're stimulating the brain, we know how many seizures they're having. We know when they take their medicines, as long as they log those. So this is work, Tori Marks, a PhD student in the lab who's done a lot of work with Dr. Kusiewicz, who's been tracking the patients. So we have, I say 23/7, because they got to spend about an hour charging the damn thing every day. So it would be nice to improve. There's still room for technology engineering improvements, right? But we detect all the seizures, and they do a free recall task. And what we've shown in this patient, again, very long time scale data. So this is not seeing a patient in the office or in the EMU for a few days. This is following a patient for 80 weeks, getting electrical brain stimulation, tracking all their seizures, and tracking their mood, or not their mood score. That's a different thing, which I'm not going to show you, but tracking how they do on this free recall task. So on the tablet, they get a list of 12 nouns. They encode those 12 nouns, and then they are asked to recall those. And this is our first patient, M1, and she's out living her life, so she's doing this at home, and randomly she gets asked to do a memory task. Not in the middle of the night. We make it during the day, allow you to have your cup of coffee. And up here is what what undergraduate students will recall, percent of words remembered. That's from this paper from Mike Kahana, and I think it was Penn undergraduates. So this is the high score for University of Pennsylvania undergraduates. You see our patient started out here. Again, it's uncommon for a patient with medial temporal lobe epilepsy. These patients have bilateral medial temporal lobe epilepsy. They're not going to have normal memory. But what we showed with this patient and it wasn't overnight, of course, over many, many weeks of adjusting stimulation and driving the seizure rate down. So it's complex. There are many things at play. What we didn't change was the medication, and what I do know is how many seizures she is having. And that's a lot to know that. But that over time, we were able to see an improvement in her memory. We can do the same thing now with mood scales. So you can do an immediate mood scale or track people's mood. So we can track sleep, we can track verbal memory, and we can track mood. And that's work that Arena Bolsakis is doing and has some nice results. I just haven't gotten them into the talk yet. I'll tell you one last thing, and this is just being analyzed now as we speak. Václav Kremen has been looking at, so we've been talking about passively recording from the brain, measuring seizures, measuring interictal spikes. What about tickling the brain and measuring the response? There's a great deal of physics behind the idea, linear response. So measuring the response of the brain. And in this network, so this is the circuit of Papes, right? We have electrodes in 2 or 3 of those nodes, the anterior nucleus of the thalamus, hippocampus, and amygdala. Can we track evoked potentials over time? And that's with the device, we've been doing that. This is data from about a month of stimulating in anterior nucleus of the thalamus, measuring in hippocampus every 15 minutes while awake and sleep, going on for days and days and days. And what Václav's shown is that there is a nice evoked response. It's very different. This shouldn't surprise you. It depends on the brain state. It depends on if you're awake or asleep. It depends on if you're in REM sleep. And it looks like if we use latency as a surrogate for excitability, and there are probably some challenges with that. Again, this is all an evolving story. But the technology is now there to track not only these passive local field potential recordings, but to actually actively ping the network and measure the response. And of course, one of the things that we'll be very interested in knowing is how this changes leading up to seizures. How this changes with the therapeutic stimulation. Can we change how the brain responds? So I'm going to end there where we are. What we see the future looks like, there's a big role, I think, for electrical stimulation therapies. You know, there are a lot of huge changes in biology advances that are just fantastic. Optogenetics, genetic engineering. My guess, I'm wrong all the time, but I'll tell you what I think. My guess is those are probably 10 years out, at least for epilepsy. I think those are 10 years out. Electrical stimulation therapies, those are there, that's possible today. So I think these are technologies where, you know, we can make a big impact with really things that are, you know, it's just engineering. We're not violating any laws of nature or even creating new things. This is just bringing to medical devices what's already existing. You know, I'll reach for my phone. All of us have our, you know, phone, and we upgrade that phone. I don't know. If Apple has their way, we do it, you know, every year or something. So this idea of an implantable with a scientific payload, getting the data off that device onto a computing platform, I think is a powerful one. You know, we need better batteries. We need better brain machine interfaces, better technology, more channels. But all of that's moving in the right direction. The idea that you could integrate that with wearables. And of course, once you get that data, you know, did the whole talk without saying artificial intelligence. So that's good. I mean, there are big changes on the horizon in computing, right? I mean, they're happening now. And so I think what is possible with integrating patients, not to sound too, you know, I don't want that to sound anyways, taking advantage of technology to create better therapies, I think, is something that's definitely possible. The idea to stimulate other networks, and important to discover what those networks are for these common problems. And again, it's not just epilepsy, so psychiatric disorders, mood disorders, memory disorders. I think we'll see opportunities in all those areas. So I'll stop there. Thanks.
Thank you so much, Greg.
I think the plan right now, well, first of all, like I will just start the discussion and we'll get into questions, but just, you know, start thinking about if you combine the two talks. like the biomarkers that of the this phenomenal experiences that Milan was talking about with what is possible technologically, and just start thinking about it. I'll just leave you at that. Because we're running down on time, we'll first take questions from the chat because we're going to have later occasion here to talk. So the first question is: do you think that the upper frequency of ripples can be actually higher than modern technology allows it to register, or are there things that the technology will not capture?
Well, I'll say an axiomatic: you won't measure it even if it occurs unless you sample. So we learned that, that's the lesson from 10 years ago, or 50, probably 20 years ago. So I don't know. There's actually, I've looked at this quite a bit recently because of Dr. Brazdil's data, and we've recorded some of these very high frequency oscillations. There's a fairly famous biophysicist, Freilich, who looked at collective modes in membranes, and because of that very high dielectric constant of membrane, speculated that there should be an oscillation, or not an oscillation, but that there could be electric field fluctuations in the megahertz range. So I'm saying I don't know. There are various theories about how microtubules coalesce. These are things I don't know anything about, but I know they've been explored. So I don't think I've answered the question. I do know that there's activity up to 1,000 hertz because I've seen it.
Okay, another question from our listeners or from the audience online. The question about the memory improvement over time thanks to stimulation. So how does it compare to memory test improvement for an average person who was not exposed to any stimuli but did the memory test?
Yeah, I think they're asking what's the learning, what's the effect of learning, and so this task is a nice task because it uses a very large database of nouns. So the learning piece, you can try this at home. Keep practicing. Neurologists do this all the time because we ask our patients, basically the number of words that you can hold in memory. It doesn't get much better, or it doesn't get better than, of course there are savants that can keep long lists, but in general it's about seven words. And you can practice your ass off for weeks and weeks and weeks, and you're still going to get about seven words. So that's my long-winded way of saying I don't think it plays much of an effect.
Okay, and we'll take a few questions from the audience for Greg specifically before we start our general discussion.
Yeah.
So thanks very much for the talk. That was very interesting and inspiring. So in one way, it's amazing how much progress we have made. At the same time, you can see just how much space there is still there and how cool are those technologies. And just, what I'm thinking was, for instance, in the context of when you think about, say, neuroprosthetics of the retina, for instance, the stuff that Zimnicki does, when you have electrodes, with the distances on, tens of microns, right? Yeah. Which, there's no way that you can have this in here. So I'm just thinking about, what is missing here, and I just wonder what is your take. So I think, to me, two things seem interesting. So one thing would be, all those interactions between those new interfaces in the brain are very much, I would say, phenomenological, in the sense, you detect the seizure and you try to respond in some sensible way, but I think they do not take into account the actual network behavior. So to me, an interesting component that I think is still missing here is to actually try to combine this with the computational neuroscience, to try to model the network activity and essentially, see how it responds, and combine this with the field propagation. Yeah. So that's one component. The second thing is, obviously, what we would like to have is even denser contacts and even smaller contacts, both for recordings and for the stimulation, right? So you could actually deliver much more precise stuff because, basically you hit with a bat, right? And you hope that you hit in the right place, right? And maybe you're lucky. And in that context also, a third question is what's your take about the Neuralink? Do you think anything useful from the perspective of the epilepsy research is going to come out of that?
Yeah. So I'll start with the last one. I've thought about that a lot. Probably now the most famous neuroscientist in the world is Elon Musk. I'm not sure that's necessarily a good thing, but that's just how the internet works. Yeah, I think I haven't used the Neuralink device to record. There's another significant advance that happened about 5 years ago with the neural pixel. So a really nice advance in technology. I see those in similar ways, the Neuralink device. Fantastic engineering, which is incredibly important because there's this big engineering gap. And I think could be really useful for, say, applications of a brain-machine interface to control a cursor on a computer or maybe a prosthetic limb. You can see I'm not saying epilepsy. I don't, so I think there's obviously what often happens, there's that technology advance that happens. I'm not sure how much thought really has even gone in to the applications of that technology to things. I know it's talked about, but when I hear the talking, I'm thinking, I'm not sure how much thought has actually gone in, because I don't think it's going to have an impact on epilepsy, to answer your question directly. I think it moves the ball forward technologically. Your high channel counts, streaming dense data off, all that's important. But I don't see a path for stimulating at the single neuron kind of level, at the microwire level. Epilepsy is not really, I mentioned, it's a disease class, I think, organized on the scale of cortical columns and populations of columns. And
it's more about synchrony.
Yeah, it's more about synchrony across the other thing that you brought up, across networks. So I think what's needed is a better continued progress in neuroscience understanding of the networks that underlie for epilepsy, but physiologic function, like how memories are encoded, the networks that are involved in that, how pathologic networks develop, whether it be diseases of psychosis, schizophrenia, mood. So there's for those diseases and for those, there's also a very large neuroscience gap. I talked a lot about there's this engineering gap for epilepsy. And that's exactly what I think you want to do. You want to do the things that you can, make a dent in, move the field forward, and in parallel, because science is, who knows how fast that's going to go, right?
Okay, we'll take another question. Maybe Kasia?
Yeah, so...
And can you rephrase, because people were...
Right, so I'll say it very succinctly, the question that there was a high false positive rate. So detections that turned out not to be seizures. And, the algorithms, you can make them better and better. It depends on the application again. So there's some applications, like the NeuroPace application, responsive neurostimulation, we've found, as many other groups that use that device, is that there's benefit to getting a lot of stimulations. And you get stimulations from detections. The idea originally was you want to detect the seizure at a very short latency before it builds up. So you want to detect it earlier and earlier. And so you make the detector hypersensitive. You maybe get 800 false positives a day, and with that you get 800 800 stimulations, and that becomes a therapy. That becomes a therapy. It depends on, so if you want to do responsive stimulation, maybe you want a lot of false positives. You want it to be hypersensitive. If you want to try and detect the seizure right at the onset. And of course, on the other hand, that rests upon the idea that there's no penalty to a false positive. It's sub-threshold electrical stimulation. We don't think that it causes, any injury or anything, so it doesn't cost you anything to do that. And in fact, we think now that that actually is beneficial because you get more therapies. Now, another application would be if you're using an algorithm for forecasting and you're going to use it to determine whether you should take more medicine that day, then there's clearly extreme consequences for false negatives. So it does depend on the application. This is an area where machine learning, convolutional neural networks and the volume of data. You can't do these things without data. The volume of data have been able to massively improve those. So yeah, I guess I'm going off on a little bit of a tangent. Now, for the FDA, I forget I think we had 1 false positive per day is what we detuned our detector to. So if you just want to have a seizure diary, not using that to drive immediate therapy, then you want it to be accurate, few false positives, but you can wait until, you don't have to detect the seizure right at the onset. You just want to say, in this 10-second block, there was a seizure. And because those local field amplitudes, become quite significant, that turns out not that hard to do.
So I have a question. What's from your opinion? What do you think how it works? I mean, how the DDS works at the neural level? And by this I mean just it's like Professor Bucic said, it's like a path that we are hitting this phenomena. And I also just wonder whether if you are able to detect it before the event, then it does not necessarily mean that in this simulation you can prevent it. Because we don't know how it works. And as you showed in one slide, many different frequencies work. Also, even lesion after the operation works for some time. Then I'm just curious, what's your opinion? I mean, how
it's a great question, and it's very important. And I like to say, I don't say what is the mechanism. Oh, yeah, I'll paraphrase it. What is the mechanism of brain stimulation? How does it work? Very important. It's not mechanism. You need an S on there. It's mechanisms. Because what I can tell you when we've looked in cell cultures, one of the first things, we started out looking at neurons just like everyone else did, and the first thing that we found, well, it wasn't the first thing that we found, but this is work Hailong Lang in the lab, that when you stimulate a culture of glial cells, you can manipulate the release of extracellular vesicles. We know that extracellular vesicles are how cells communicate, one of the mechanisms by which cells communicate. They have microRNA. There are these nano-sized particles that go from one bleb off one membrane and then fuse with another membrane. We demonstrated in the lab that you can actually manipulate the cargo in those extracellular vesicles. You can change the microRNA that's in that extracellular vesicles, and you can increase and modulate the number of those extracellular vesicles. I suspect that that's one mechanism that doesn't involve neurons at all, doesn't involve action potentials, and is a molecular mechanism of exosome communication between glial cells. So that's one mechanism that we recently described, and of course, well, we described it, we published it, and then we wrote a grant on it. And so we're actively looking at these non-neuronal mechanisms of electrical stimulation. And there's, you know, I think this is a really interesting idea because all organisms evolved within this milieu of electrical fields. And so the idea to think that how cells communicate and are affected by electrical fields, there's probably, obviously there are other mechanisms. And then, you know, most of the work is focused on neurons. And of course, you can do high frequency stimulation and create a depolarization block and stop neurons from firing. And that was the early observation of I don't know if he's the first, but Binabed, you know, they discovered that you could treat tremor movement disorders with lesions. And so it became, how could you create a functional lesion? And that approach, you know, piggybacked on the neuroscience that high frequency stimulation can create a depolarization block of neurons. Whether that is the mechanism, I doubt that that's the mechanism by which epilepsy primarily works. Again, I'm sure it's one of the mechanisms, but yeah, so I haven't answered your question, but I've acknowledged your question as an important one and told you some of the things that are happening. The other, I'll go out on a limb here. I think another area that's so it's not all about the neurons. And 5 years ago, I was all excited about the glia. It's not all about the glia either. So the extracellular space. So the extracellular space that has to be, I said the brain's not a bag of saline. The extracellular space that has to be maintained so that neurons can do what they do is a very intricately controlled area. And there are many proteins and charged bound particles that get, I'm going to guess that brain stimulation modulates also the extracellular space, that space between neurons and glia. And so, yeah, mechanisms of brain stimulation. And then, of course, it depends on where you stimulate. I mean, the anterior nucleus of the thalamus, one thing that and I don't know that this is the mechanism by the anti-epileptic effect is, but if you stimulate the anterior nucleus of the thalamus, we can really suppress the activity in the hippocampus as part of that circuit of Papes. And, you know, that's been shown by other groups, and some early Medtronic work in sheep actually showed that. So that's a direct, I suspect, direct neuronal effect, but I'm not, you know, what role that actually plays in suppressing seizures, I'm not sure. So.
I have the last question.
To Greg specifically or a general one? Okay.
So he's going to ask a hard question.
Exciting and literally stimulating talk. My question will be crazy and provocative.
All right, I like
that.
One of the programs in epilepsy patients with the stimulation and we discussed is recharging.
Yeah.
You've got a sensing electrode in the brain.
Yeah.
There is a lot of energy.
Yeah.
Will it be used for recharging?
Yeah. People are looking at this. I don't know how effective it's been, but I suspect, I mean, the brain, of course, generates an enormous amount of energy, and I mean, obviously all the electrical activity simplistically is built upon the idea of a battery dissociating concentrations of ions. So the brain is very, very good at doing that. And yeah, can you harvest energy from the brain, electrical energy? How you would have to do that is, you know, separate potassium and sodium and charge an external device. I suspect that will happen someday. I don't know if it'll be.
Well, but that begins to sound like the vision from Matrix, that the humans are powering computers, to do their work. That's why they're provocative, I guess.
Yeah, the idea, the integration of humans and machines. It has kind of a negative sound to it, but I think about the very positive potential for that.
And there's a whole discipline called cybernetics, which is just that, right?
Yeah.
Great. So with that question, I think we'll enter into a grand discussion. So I'll invite Professor Brazdil and Professor Worrell to either sit, because you've been standing a lot, or just stand here so that we can see you on the camera. And then we will start a discussion. I'll just throw questions, and either of you will answer them. And we'll start with.
Keep standing, or should we sit?
So it will be great if we could see you too. So the camera is kind of,
all right.
Having the central view. So I'll start with a provocative and very general, just to get a little bit down from kind of physiology and those nomenclature that maybe not everyone is very familiar with, especially on the other side of the screen. So the question is kind of going along the Neuralink maybe ideas and some of this crazy kind of machine and human interfaces of what is possible. So we've heard what is possible clinically, but the question is there any research done on how to write some information into brain and skipping human senses? Write some information into the brain and skip human senses. So, it's a general question, but maybe, you've got some ideas or speculations.
I don't think so, of course. So we can support memory consolidation, which was already mentioned by Greg, using the stimulation of hippocampus. But this is something which very likely cannot be used in the clinical practice. So I don't think so.
Yeah, I mean, I can think of angles on that where there are elements of that. Studies that have shown that you can change someone's perception by stimulation. So there maybe you're changing what's being written into the brain. You're not like writing in the brain, but you're manipulating those. But yeah, I know that's.
The microphone, sorry, we cannot hear you. Well, and the Neuralink, I think it's more not about stimulating or writing in, but definitely reading information from the contents of the mind, for example. I think that's the one that goes into Neuralink.
You mean reading our minds?
Yeah, reading minds, yeah.
Oh.
What about that?
Yeah.
I'll leave that alone.
There are, as far as I'm aware, there were performed many experiments using functional MRI, for example, to find out what's happening in our brain. And of course, to some degree, we are already able, even online, there is MRI feedback performed, including our core facilities performing experiments with fMRI. Feedback, which is a crazy idea, but the machine can differentiate something from our minds already. And the question is, however, to some degree it will be possible, and to what details these techniques will be able to read our minds. I hope that just to some degree.
Okay, we can take a question maybe from the audience now.
So I have a question. Do you see any possibility to use less invasive methods of stimulation, like EMS, for example, for stimulation of this seizures, right? Memory, or maybe to use vessels as well, like in the Professor Oxley
very active area, you know, non-invasive stimulation and transcranial magnetic stimulation. And, you know, as you probably know, it's already, from a medical point of view, it's approved for depression. It's not well understood how it works. So back to that, I think, you know, back to that idea that sometimes there are engineering gaps. We should fill those immediately, you know. I always say there's no excuse for engineering gaps. If something can be done, that should be done. There are, I think, significant science gaps, but, you know, people certainly working on that and emerging understanding. And so, yes, I think those less invasive techniques will be possible. I mean, you know, can you stimulate, say, deep structures, say, you know, the CL nucleus of the thalamus in a very controlled fashion using, you know, an external transcranial magnetic stimulation? Probably not, but you could use ultrasound. You know, you can target ultrasound to create lesions, or if you wanted to just heat up an area, say, centrolaminar nucleus of the thalamus, you know, below the level at which injury happens, so that would make that region more excitable. No question you could do that. So I think.
Yeah, frankly, there were many experiments done with transcranial magnetic stimulation as a treatment method in epileptic patients, but in general, these methods didn't show to be very efficacious. And what's also, and this is probably the most important, is that if there is any effect, then this effect is transitory. Another issue is that with transcranial magnetic stimulation, as Greg already said, you are unable to stimulate the deep brain structures. It would be possible to stimulate, for example, cerebellum, which was done also with depth electrodes, but again, the accessibility for transcranial magnetic stimulation of cerebellum is quite complicated. So I think that this probably is not the future of the epilepsy treatment, but it's just my personal experience.
Yeah, so this will be me specific. So you can definitely, and we've done it and published it, that you can actually record high frequency oscillations from within the vessels. We've done that. So the recording part of it, and I lost a bet on that, so I remember it very well. Because I thought it's a highly conductive medium. I figured all the currents would be shunted away and you wouldn't, but you clearly can get nice recordings from inside the vessel. Now, yeah, so that's a fact. What you can use it for, stimulation would be one possibility. I think, yeah, that's a.
On the other hand, the problem
there's information in the brain that kind of doesn't reach maybe the conscious or the awareness, or you don't have access to this. Mm-hmm. So the question is about the actually somatic marker hypothesis of Antonio Damasio, and the question is that this is a very interesting model, quite similar to the mechanisms you described in your answer, and in short, when a subject is resolving cognitive tasks in stressful conditions or data overload, decision making is subconsciously, could the decision making be subconsciously delegated to like ventromedial prefrontal cortex or the amygdala or other structures, and kind of made subconsciously, and then you reach a conscious decision. Like there's a lot of autonomic and other subconscious processes that contribute that you're not aware of, and then
definitely I would say that the majority of our cognitive processes are very likely ongoing unconsciously, and only a small part of all the mental processing is going consciously. Unconscious and conscious, it goes hand with hand. So it makes sense that that when we focus our attention on some task and there is something happening in the surrounding, we don't pay attention to these other issues, and doesn't mean that these information are not coming to our brain and that these information are processing in the brain and even on a quite high level. So yes, that's the reason I guess that our mind is so complicated, because there is a lot of information of which we have no clues that there are. That's my impression.
And then could we use this information like to predict some things and help the brain, in therapies?
Probably yes.
Do you think that DBS can be transferred to other psychiatric diseases and used for that before? I know that it's already used for some cases for depression, but also there are some trials for schizophrenia, and whether it will take out the medication, so also prediction for the future.
Well, there are a few examples. So it's already stimulation anterior limb of the internal capsule approved for obsessive compulsive disease, an incredibly disabling disease. So that's certainly one application. And I think we'll see. We will see more. I think, again, this is one where there are science gaps. The technology for stimulating is there, but the networks underlying some of these, diseases of the mind. That's why epilepsy is incredibly complicated. And I always say, boy, this is a tough disease. But psychiatric disease, yeah. I mean, it's one, you don't have we don't have clear biomarkers. But all that said, it's a frontier, definitely, and I suspect we will see more applications. It was if you will, depressing that the brain stimulation for severe depression was not positive, and that's a little bit of the fact is actually there was a signal there. It just wasn't as robust as industry drivers wanted it to be. So there was definitely there was something there. So that's another, obviously depression impacts many people, and it's comorbid with many other diseases. So I suspect, I'm going to guess that's going to be one where we'll see progress in the 5-10 years.
Definitely, yes. I absolutely agree. I'm not a psychiatrist and have no good latest review in this field. But in the field of vagal nerve stimulation, people are trying to use these stimulation techniques for many diseases, including stroke, for example, which seems to be crazy, and the treatment of obesity, etc. So I'm sure that also in the field of DBS in psychiatry, there is very strong research.
I guess the joke is there. Milan says, I'm not a psychiatrist, but he is married to one.
Yes, yes, my wife is.
So another last question from the audience online. The question is about seizures, but I think we can go further and say about events in general. So could this brain stimulation technology be used to trigger controllable or small events so that we learn something about how they're generated, and therefore how can we mitigate them, or how can we control them or treat them?
Yeah. No, that's one thing joggled my memory with that. The other thing that we're in a position now to do, historically most of the brain research is you have someone performing a task and you measure their brain activity. We're now in a position where we can actually measure activity and then trigger the task or trigger the question. So if you think mood, say, is tied to amygdala hippocampal coupling, well, you can measure that continuously, and that's going to be distributed probably in a very non-normal distribution, and when you're in those long tail out there on the tail with strong or weak coupling, you can start querying the patient in an automated way through the technology. So that's one way that we're using signals in the brain then to trigger querying subjects. And then I think an extension of that would be, electrical stimulation and querying the patient as to, what effects. And of course, electrical stimulation is used for mapping epileptic networks currently.
Exactly. Yeah. It's just some kind of evoked potentials. Yeah. Cortical evoked potentials, for example. Yeah.
Any more questions from the audience here?
I was always wondering how the systems about brain simulations are protected from hacking, like autonomous cars or something.
Yeah.
Because, it might be a case like, okay, give me $100, or otherwise you're not going to be sleeping, for the next 2 weeks or something.
Yeah, no.
Can we paraphrase it?
Right, so ask a very, very important question about hacking the brain. So if you have a device, we can extend that to how do we protect the information, the public health, the human public information. Those are very important questions. There's a lot of technology for encoding. Swiss banks send billions of dollars around the world electronically all day long. So there's a great deal of technology built around encoding and doing that, safely. That doesn't completely mitigate or eliminate the risk of hacking, obviously. So it's a really important question. For brain stimulation, the approach that is, I think, you'll see that will be taken is only with a direct connection with the device and the physician as someone's sitting down can you change the parameters around which you can stimulate. So you set, if you will, set the guardrails, and you would want to test that face to face to make sure that's a safe stimulation paradigm. So in that way, if the device were hacked, it wouldn't be able to drive into stimulation into an unsafe, stimulation range. But we haven't talked, and I was neglectful. I didn't have the ethics of all of this is hugely important, right? And it's not only the question, can we do things, but should we do things? And there's the bioethics of brain stimulation and how this could potentially affect a person's agency, their sense of self, who they are. Those are very important questions that are balanced against trying to create new therapies for terrible diseases. So yeah, to answer your question, how do you keep people from hacking devices? Well, you put a lot of effort into making secure technologies, and then do the risk strategy of, well, if that happens, what is the consequence of that? And if you can't reduce that risk, then that shouldn't be a technology that's deployed. If it's not an acceptable risk, for instance, what's the risk of someone being able to control the parameters? Well, if the risk of that is too high, then you have to make a judgment on that.
The question is, what would happen if somebody will hack the system and will increase, for example, the stimulation parameters up to, let's say, continuous stimulation, and whether or not there will be a damage of the anterior thalamic nuclei, because this similar situation is with vagal nerve, VNS, when we know that there is some maximum stimulation, about 1 to 2 non and stimulation and off-time stimulation, and after that probably can cause an injury of the nerve. Yeah, so I think that it's extremely important, but I suspect that it probably will increase the price of the system to prevent this hacking and use these Swiss patents how to prevent them.
And again, you know, it is quite well understood what the parameters are that of stimulation that would cause injury, how much, you know, whether it's, you know, common 30 microcoulombs per centimeter squared. You know, you keep the therapy below that, and you can build the device, you can set the parameters on the device so that it just simply cannot go beyond that. So that's the current approach that we've had with our devices that are out in the field. So even if someone gained control of it, the device just simply doesn't have that in the parameter space. It goes up to 20 microcoulombs per centimeter squared. But it's a good question and one that has to continually be worked on.
There was an interesting comment about writing information or simply inducing phenomena in the brain stimulation, for example. I know a lot of the stimulation is obviously a threshold, so patients are not aware that they are stimulated.
Yeah.
But in theory, if it was supra-threshold, then there's a comment and question about how would it compare, would it feel real? You know, would it be like an illusion or hallucination, or, you know, what are your comments?
There could be stimulated almost everything, actually. Out-of-body experience is one phenomenon which is extremely interesting, and this was repeatedly stimulated when the electrical, using depth electrodes implanted into the supramarginal gyrus on the right side in 1 or 2 patients already, and this very eerie phenomenon was stimulated in the epileptic patients. Déjà vu key and all these memory illusions were stimulated. You can stimulate some movement, some some sensations, etc. So, but we are doing this stimulation as a part of the preoperative protocol to either provoke the epileptic seizure, which is very important to clearly identify the epileptogenic zone or epileptic focus, and also to map the cortex to be sure that the subsequent surgery will not result in some functional deficit which is unacceptable in epilepsy surgery.
Okay, final question?
I'd like to ask about biocompatibility of the materials. It is real challenge right now because when we increase the number of these devices around in a society, then we have a problem that might happen the increasing number of the infection. How today materials comply with the requirements of these purposes? There is any problems with the calcination of the infection or any other problems?
Yeah, I mean, certainly infection, you know, a foreign body. Is the surgeons in the audience, you know this better than anyone. Yeah, it's always going to be a risk. And, you know, biocompatibility, I think there's, if you will, a suite of materials that are well established. Going outside those, while there may have some advantages, the regulatory challenges of that are huge. So for instance, all the electrodes, the business end of the electrodes are platinum iridium electrodes. And you know, it's hard to do anything other than that. You know, sometimes, of course, because they're, you know, you're not going to use a copper electrode because of the oxidative, but so as long as they're platinum or iridium, for example, you know, that's that's a clean material for stimulating the brain. Most of the cans are titanium. And you know, all that technology that goes into making something hermetically sealed is not trivial at all. And then whether they be, you know, I think largely silicone substrate for the flexible substrates. Yeah, it's a very active area, you know, to make materials lighter, more flexible, to conform more easily to the brain. All of those are, you know, areas of, as you I'm sure know, you know, active areas of research, and probably going to have significant impact, you know, over the future as better materials get created.
I think so. One of the problem which we faced too also, and we are not the only one, that there might be an allergy for these depth electrodes. And we've got one patient and 2 patients were in Prague in Motol. And with the allergy it's a problem because in patients it's developed the brain edema. And definitely you have to remove the electrodes as quickly as possible. So the investigation is over in this moment. So it's problematic, but this might happen, and I would say that the biocompatibility is not 100%.
All right. So with that, I would like to thank very much our speakers today for an excellent and stimulating discussion. Thank you. I would like to also thank distinguished guests who are here in the audience who have contributed to this discussion with questions. And I'm sure there's going to be follow-up discussions offline. And I'd like to thank also all the audience through online who are watching. Yes, thank you so much.
Thank you.
Thank you. So now.